All right, awesome. So my name is Prabal Saxena, and it's great to be back here to give a talk about the moon. So this is my, I have to get Frank Sinatra in there a little bit too, so that's why I put "Fly Me to the Moon" whenever I want to talk about this topic. But really, Stacey did a great job describing what it'll be about. It's really why humans may not be the only life on the lunar surface in the future. And one of the reasons I'm very excited about giving this talk, and I think Stacey probably is pretty excited as well, is that we're maybe a little bit under 2 weeks from when Artemis 2 is going to have its first window for launch. So, for those who might not know, Artemis 2 is going to be the next mission to the moon. It is planned to carry four astronauts uh, on a trip basically around the moon, and they will be the first humans to directly see the far side of the moon. So, not the dark side of the moon. I know we're all Pink Floyd fans, but they will be the first ones who will be able to see that. And it's going to actually test a number of really important engineering milestones that are going to be important for the return of humans to the moon and a more sustainable presence on that body. But they'll also be able to do really interesting science. So I encourage you to go ahead and, when you can, look at resources that I know NASA's been putting a lot out in terms of what Artemis 2 will include in terms of science, in terms of a lot of the really exciting things that the astronauts there will do. So that's kind of sets the scene for this exploration for the moon. And when I think about this particular mission, they're going to be they're going to be traveling around the moon and they'll get to see it through a window. But Artemis 3, the idea is that they'll land, and all other future missions, there will be a sustainable presence on the surface. So I want you to put yourself in that perspective. Think about, hey, if I was an astronaut, if I was going to go to the moon and I was planning to then be there for a little while, if I was going to be able to explore, look at a lot of the exciting things there, which I'll talk about because we'll be going to a very different region of the moon than we have been to before. What would you want? What would you be excited about? And one of the things I always think about, and I talked to Stacy about once, I asked, who would you want to take with you to the moon? And for me, I have a very obvious answer, and that's these guys. And while it would be awesome to take Buddy, Biscuit, and all these guys to the moon, they would probably kind of be not as well behaved. It'd be harder to find a spacesuit. Unfortunately, and both fortunately, the things that we almost certainly will take to the moon are what Stacy described in that introduction and was in the title. Instead of our awesome pets, we will probably take microbial life. Um, so when I say microbial life, I'm really talking about anything that is basically not visible with your naked eye. So things that are really, really small. So these include bacteria, fungi, and viruses. And one of the questions that you might first ask is, well, all right, that's great. We are going to take a bunch of these things to the moon. Probably wants to take pets to the moon. Why are we going? Why are we going back to the moon? Some of you may remember kind of the exploration during the Apollo era. So we've been to the moon before, right? We've been to the moon 6 times where astronauts have actually landed and explored and hit golf balls and taken samples and done really wonderful experiments and learned about this really great preservation of what has happened in our solar system. So the moon is this wonderful record of what has happened in the past, how our solar system formed, how it has evolved over time, and even how kind of the Earth has evolved over time. So these are the different landing sites that the Apollo missions went to. And one thing, hopefully, that you'll notice really quickly is one, these are all on the near side. So these are on the side that if you look up at the night sky, you can see that particular portion of the moon. And then the other thing is they're all kind of equatorial. So they're all kind of close to the middle of the moon. So the moon, as opposed to the Earth, where the Earth has kind of a tilt because of its obliquity, the moon is not as tilted. So because it doesn't have that tilt, you're really looking pretty much flat on here relatively when you account for our tilt. And these are all landing sites that are within about 35 degrees from the equator. So you might think, all right, well, why is that important? It turns out that because the moon doesn't have an atmosphere, the conditions on those particular regions are very different versus if you go to the poles of the moon. And that's something we've learned because we've had a lot of other missions that have been uncrewed in the past since the Apollo missions. In fact, the cadence at which we've been sending missions, the cadence at which humans have been exploring the moon has went up significantly in the last couple of decades. So within the last 5 years, they've been over at this point, 22 is out of date. It's something like 25 missions to the moon. And we now, you've probably seen in the news, have had private companies that have been basically transporting instruments and trying to land on the moon as well with, varying degrees of success. So one of the reasons we want to go to the moon is because even though we've been to certain portions of the moon, we actually haven't really explored the kind of diverse range of environments on the moon. And some of those environments are really exciting because they have very different conditions on those polar regions versus the surface. So I'll talk a little bit more about that in a second, but those regions that we want to go to, these polar regions, we think that we can actually basically have a more sustainable presence there. And that's kind of the plan behind the Artemis campaign. This is kind of a depiction of what eventually we would envision Artemis being able to have up on the surface near a landing site. So the idea is that you'd have a lander, you'd have a pressurized rover, which you see in the middle there, that would allow astronauts to really take long road trips so they could visit their favorite crater or, geological feature or whatever else they found really interesting. And that they'd be able to conduct not only a bunch of science, but also understanding of how to live on another planetary body. So that's kind of the long-term vision of what the Artemis program is about. And besides returning humans to the moon and establishing this long-term presence, a really important motivation behind this Artemis program is that it's going to serve as a proving ground for Mars exploration. So Mars is really exciting from my perspective as a scientist because it might be the best chance we'll have to find evidence of life that existed either in the past or currently exists in our solar system outside of the Earth. So there's pretty significant evidence that Mars might have had flowing water in the not too far future, significant amounts of flowing water in kind of the ancient past. And that there might have been times when, by our conventions of what we think are conditions that are suitable for life, that Mars might have actually had some of those conditions. So this kind of sets, I think, why we're going to the moon. So we're going to the moon because one, inherently, it has a ton of really interesting scientific work. The moon has all this information that it tells us about how the solar system evolved, how the Earth changed over time, how our galaxy changed over time, and also how our sun changed over time. It also helps us learn how to explore space and how to survive on another planetary body and have a long-term presence there. And then finally, it also really lets us serve as a proving ground for Mars exploration, where there are a lot of other considerations we'll have to have about successfully exploring that particular planetary body. So Artemis 2 is the one that I kind of led off with. So that's the one that'll launch in a couple of weeks, hopefully. And Artemis 3 is the next mission that's planned after that. And Artemis 3 is the first mission where humans will go ahead and land on the moon again. And in this particular case, the plan, as you can see in this inset right here with these yellow squares, Artemis 3 will land in one of these landing sites. These are the Artemis 3 candidate regions. And you will have astronauts go ahead and explore the South Pole region. So this is a picture of the South Pole region here. And you can see a bunch of, one of the things that's really interesting here is you can see some of these craters and they're really dark. And that is not, artistic like discretion here. Those are actually how the craters look. Because the moon is not tilted and because you actually have topography, because you have these craters that have rims that go up and depressions in the middle, some of these insides of the craters never see sunlight. And because you have no atmosphere, that means there's no way to redistribute heat. So the temperatures in the middle of some of these craters, which are often referred to as permanently shadowed regions, they end up being incredibly cold. So way colder than anything we typically see in our daily life. We're talking like negative 200 degrees Celsius, and in some cases even colder. Now, the exciting thing that those craters can potentially offer is that when you get to those temperatures, even if you have a vacuum on the surface, which is essentially what the lunar surface has instead of an atmosphere, you can have water ice both on the surface and mixed in with the lunar soil that is underneath. And that's really exciting for people who want to explore the moon because liquid water and water in general is just one of the most precious resources in terms of enabling exploration. It also gives you a lot of really interesting scientific information because it tells you how water and other things that can freeze and evaporate and sublimate and kind of transport, how those things have changed over time, have they been kept in time. So Artemis 3 offers to be really an amazing scientific exploration and engineering mission. And it's because of the place we're going to on the moon, which is very different from the places we've been to before. So, right. So that's the reason why we're going to the moon, why Artemis is something NASA is so focused on. And I love the Artemis program. I think I love the idea of going back to the moon. There's so much cool science we can do. Please put in questions. I'm happy to answer those, even if it's not related to microbes. But that kind of sets the stage for why we're going to the moon and why we're going to this specific place. Now, this runs into the other thing. So when I talked about microbes, the reason we're talking about microbes is because humans are messy. And humans are messy not just because we have food fights, as you can see in here, or we have dubious hygiene sometimes, depending. I just went for a run. If I didn't shower, I'm sure I'd be in that case too. But humans are also just naturally messy from the perspective of traveling to space and thinking about unintended transfer of life. So one of the things that people do and that organizations do as we are planning trips to space, as we're planning to send things into space, is we try to avoid transfer of life and organics that we don't intend to send somewhere. So what you do is you put things together, you build things in what are called clean rooms. Clean rooms are often heavily filtered, and in order to make sure that you're not bringing along things, you're not kind of contaminating instruments or a spacecraft that you plan to send somewhere. And in addition to kind of filtering things, they also actively do things to try to make sure that you're killing off any of these microbes that might be transferred. So there's a bunch of different techniques that they use to try to kind of kill off microbes that might be on an instrument or on a spacecraft. This can include things that it's kind of intuitive. So heating things up, using UV light, using kind of like things like disinfectants. And that all works somewhat well for these spacecraft and instruments. But one of the tricky things is that humans, when you're trying to send humans to space, you cannot use the same clean room techniques. You can't use the same techniques to try to get rid of this additional life. And part of that reason is when I said humans are messy, we at all times have millions of hitchhikers in our body that are not just human cells. And that is because humans carry trillions of bacteria in their body and between 10,000 to a million microbes per square centimeter on your skin, depending on the study. So usually when I've told kind of gone to schools and told people that, kids are like, what is going on? This is gross. It's not that scary. The skin does a really great job of protecting you from things that might be dangerous. So you can kind of see here a picture of the skin and the different, basically, the structure that kind of protects from the things that are right on the surface. But those bacteria, those fungi, those things are still on the surface of your skin. They're still hitching a ride with you. The same is true of inside of your body as well. So I'm sure if you're eating yogurt, you're looking for probiotic, yogurt, probiotics that'll help your gut, your stomach, all that other stuff. And it turns out our body has microbes that are inside our body that often help us in some cases that are not necessarily dangerous to us. And we have them in a lot of different places. And they're often not only a ton of them, they're often really diverse too in terms of types of species. So that's particularly interesting because one, we have all these hitchhikers naturally on us. And that means that those 4 astronauts who this time are going to be flying around the moon, but then in a future mission would then land and explore the surface, will all inherently be carrying these hitchhikers around with them. And we've seen this before. So if you look at the International Space Station, astronauts continuously go to the International Space Station. You have astronauts constantly going there, coming back, and they are humans on there, right? So they are you can see them here eating pizza. They're eating a bunch of things. They are also going to the bathroom. They're going when nature calls, and they're also exercising. So they're doing normal human things, and whenever we do normal human things, we are transferring some of those microbes that exist on our body to the surfaces and environments around us. So within the ISS interior, every time you're brushing up against something, you might be leaving some type of microbe on that particular surface. Every time you cough, as we kind of know from respiratory viruses and things like that we can transfer to each other, you're often giving off things that were inside your body. with respect to microbial life. And then if let's say you're getting a really good workout, you might be breathing especially heavy, you might cough, you might be touching a surface or something like that. As you can see, riding a bike or running in 0G means you need to be kind of stable. You need to have something to hold yourself on there so you're not just flying around. And in those cases, there's multiple ways where you could be transferring microbial life to kind of the nearby environment. And in fact, in the ISS, they actually found that this was happening really, really often. So this is an area that was right near an exercise area. And what the astronauts here would do is they would put their towels after they just had a workout and they would hang them here. And that black stuff you see there is actually Aspergillus fungi. So Aspergillus is something you see around. It's a very common fungi. And because of that dampness, there are all these spores that were just hanging out there and they're just waiting for the right conditions and they started to grow. So even within a place, and you can imagine, we try to make sure the ISS and everybody else who goes there, they go through all these procedures that try to reduce that type of unintended microbial life transfer. But this is kind of where we get to humans being messy. You are inherently going to be giving a ride to a bunch of these different microbes. So one of the interesting things is we've talked about how humans will bring stuff. You might have this question, well, okay, that's great, Prabal. Humans might be bringing all this microbial life. Why does it matter in space? The inside of the ISS is not the same as space or the surface of the moon. And you're absolutely right. So the inside of the ISS, the inside of a cabin of something that lands on the moon, or the room you might be watching this talk from right now, you probably have a much nicer temperature in there. It's probably not too hot or cold. You hopefully have an atmosphere that is enabling you to breathe in some oxygen. So you have all these conditions that not only do we find pretty good, but a lot of the microbial life that we're giving a ride to also probably finds to be a pretty sweet spot. But one of the things that's been really fascinating over the last 30 years, there have been a number of studies that have examined, can microbes survive in space-like conditions? And the answer is a resounding yes. So microbes have been found to survive space-like conditions in both experiments, also in purposeful exposure to space on missions that have just said, hey, let's expose these microbes to the open vacuum of space and also to some of the conditions in low Earth orbit. And then also in unintended cases where they found on some exterior surfaces of the International Space Station, they found that once they take those back, swab them, they found microbes that have still survived. And the crazy thing is these microbes are not all just like the champions of surviving and are really extreme. A lot of these microbes that they found that have survived are really common ones. So Aspergillus, for example, that's something that we just saw was on the inside of the space station. That is a microbe that people have found can survive a lot of these conditions. But also other things, other bacteria, other fungi that we see every day that exists in the soil, that probably exists in your room with you, or certainly in my room with me right now. A lot of those things have been found to survive when exposed to not only space-like conditions, but also different portions of space.
Hello,
So,
can I interrupt
yep,
you for a second?
yeah,
We have a
absolutely.
question. Have we already contaminated the moon with the Apollo mission?
This is an awesome, awesome question. Okay, so Stacy always worries because I'm probably on the goofier side of scientists. So I will tell you about another scientist who I work with who is slightly goofier and who just to answer that question. So when the astronauts went to the moon during the Apollo missions, what they actually did was instead of going to an airlock, so right now a lot of the ways that we think about exploring the moon is you say astronauts go in a cabin and then they have an airlock right next to them. And what they do is they go into that airlock, they get in their suits, you pump out the air from that airlock once you close the doors to the cabin, and then you open the door and they can go out. And they do it safely so you don't get the bend. So the pressure changes at a slow rate. In Apollo, they had no airlock. So what they did was they just said, that cabin, we're going to go ahead and slowly open the door and pump out the air into the exterior. So everything that was floating around in there, everything that was still on the surface that hadn't been picked up by a filter, almost certainly went out there. Now, the other thing, the part that I'm gonna call out the scientist Jason Dworkin. He tells really bad jokes. So he would come to me, and he was the one of the lead scientists on the OREX mission, where they did that little scoop of an asteroid and they were looking for organics. So a really wonderful mission. I encourage you to look up. He would come up to me and be like, Prabal, my number 2 priority on the moon is to get the poop bags that the astronauts left from Apollo. So after I groaned, that is actually the main answer for have we already transferred microbes to the moon? Yes, for sure. And the reason I can tell you that with certainty is because the astronauts, when they did, when nature called, they actually took their waste, they had it in bags, and they put it underneath the landers that were there. So those bags that have contained kind of the things that the astronauts left there, those are a fascinating biological experiment at this point. So those, even though they were in a bag, which is somewhat sealed but somewhat permeable, those are microbial. That's now microbial life that has basically existed on the lunar surface for something over 50 years, exposed to a very different radiation environment, exposed to temperature range like fluctuations that are pretty extreme, and that really exists in unknown environment with respect to how it's changed over time. So we've definitely already contaminated, but so far, because they were under these landers, and there's no evidence that they've been moved or anything like that. While we probably contaminated those local areas, it's not clear that we've contaminated kind of other regions. And this is actually a really great question that flows into something else I want to talk about. So while we might have sent that life there at that particular time, one of the things is, can those microbes survive? And the regions we went to on the moon, I'm just going to scroll back really quickly. The regions we went to on the moon during Apollo were those equatorial regions. And those equatorial regions have a pretty normal day-night cycle. So they pretty much have something like half daytime, half nighttime. And because there's no atmosphere to smooth out temperature variations, during the night, it ends up being super cold. It ends up being like negative 200 degrees Celsius. And during the day, it ends up being incredibly hot. So you get to easily over 100 degrees Celsius, and you get to really, really hot temperatures that will typically be thought of as killing off most of the microbes that are exposed to that vacuum. So if they're just completely exposed on the surface, if something actually happened to land and they happen to land in these areas, you would think that basically the cycle of really, really high temperatures, that the tons of UV light that they're exposed to would probably kill them. Now, that's not the same story for the region we're planning to go to now. And the way to think about that is through this, what actually kills some of these microbes. So the 2 best things that kill microbes, there have been a couple studies that have looked at this from the perspective of space. Obviously, there's lots of studies that have looked at this from the health perspective, are ultraviolet light, which is the most effective way of killing a lot of these microbes once you get above a certain threshold, and high temperatures. And this should be pretty familiar to a lot of you. I mean, if you think about pasteurization, how you kind of boil things to kill off anything that might be kind of dangerous, that's something we do on an everyday basis. UV light, I think we all know, is also something that's used to kill off unwanted microbes. And then there's other ways you can also potentially kill off some of these microbes, but they tend to be less effective. So energetic particle radiation does a good job, but it depends on the density, so how much radiation you're sending, and basically how dense your microbes are. So if your microbes are spread around somewhere, but you don't have a ton of radiation that's hitting them, they might kill some of the microbes, but they won't kill all of them. And it turns out vacuum, the exposure to things that lack an atmosphere, basically don't really do an effective job of killing microbes. The funny thing is that the cold temperatures on the lunar poles, for example, and vacuum, when you combine them, almost can help some of those microbes survive better. It can help them basically get in this hardy state where they say, all right, I'm closing shop. I'm going to go ahead and not metabolize stuff as much, not do my normal things, but I'm going to become freeze-dried and kind of survive more effectively. So vacuum can kill microbes to a small extent, but in some cases it actually has no effect or even kind of a positive effect, the same way that cold temperatures do. And this is kind of underpinned by a lot of studies that have studied different types of microbes and how they respond to these different types of ways of trying to kill them. So why is this relevant? I've kind of alluded to this, but because the moon does not have that tilt the same way the Earth does. The polar regions on our planet, the polar regions are colder. On the moon, because the sun never gets that high at these high latitudes, at these polar regions, you basically have really, really cold temperatures and a lack of sunlight. So whereas on, let's say, the North Pole, because the sun is tilted 23.5 degrees, in the middle of summer, if you're right at the North Pole, you will see the sun at 23.5 degrees in the sky. But on the moon, if you're right at the pole, because the um, tilt of the moon relative to the same way that you think about the Earth is only about 1.5 degrees, the moon would only get up to about 1.5 degrees in that sky, which means it'd be basically right on the horizon. So you'd never really see it get above that horizon. So one, that's a pretty weird scenario to be in. But importantly, for those things that we just discussed, what does an effective job of killing microbes? One, the temperatures on the lunar South Pole are extremely cold. So if you look at basically what lighting looks like on the surface of the South Pole, there are massive shadows everywhere. That's partially why we have those permanently shadowed craters too, those regions where basically the walls of the crater block any light out. So because of that, you end up having really, really cold temperatures, which, like we said, can sometimes help preserve life that falls out there. It also means you basically never have really, really high temperatures. You never get those temperatures that might kill some of the microbes we're interested in. The other thing that's really interesting is that the other thing that does a really good job of killing microbial life is ultraviolet light. And your main source of ultraviolet light for the lunar surface is going to be the sun. So again, you have significantly reduced ultraviolet light to the point where you might not be able to actually have enough to kill a microbe that might land on the surface. And there's some pretty good examples right here in this picture that are underneath that cartoon. So this cartoon kind of shows how the sun never gets that high. So if you have even a little bit of topography, if you have a little bit of a crater or a little bit of a hill, it can actually block light and you can have portions that never get any sunlight, never get any ultraviolet light directly. And you can kind of see that in these pictures from missions that have landed closer to the polar regions. You can see these little craters and they're pretty small scale. So some of these are only a meter across, some of them are even smaller. And these are real, those dark places are real portions that never see sunlight. So they're absolutely these places that you would think might be able to be survivable for a microbe that might be able to find its way in there. The other kind of interesting thing is if you look at that picture of the astronaut by that boulder, this is a more equatorial region, but you can still see there's boulders, any type of topography can provide some of the shadowing that also keeps you away from the sun. So this kind of gives you a sense of why the polar regions on the moon are so different than these equatorial regions. They just have a completely different environment that has a very different relationship with the sun, and as a result, has very different surface properties. And that makes it really tricky for astronauts to explore because then you have to deal with basically exploring in this really big freezer that's not really well lit. But it also means that microbes that might be able to find their way there might actually be able to survive. So this is a very busy picture, and I'm just going to talk about kind of the general things you can kind of look at this. So this is, again, fresh hot off the presses. It should be hopefully be published in a month or so, and you might see it in the news then. But I have a bunch of colleagues, a bunch of astrobiologists, biochemists, lunar scientist friends who I worked with. And what we did is we said, all right, we kind of know what the topography of the surface of the moon is. We know the places we want to go to, the lunar South Pole. And the lunar North Pole also has similar kind of a similar environment. So can we model how much ultraviolet light different portions of that polar region receive and how the temperatures kind of vary? And then can we compare the temperatures and the ultraviolet light in each of those portions of the South Pole and use data for five really common microbes. And I would say four very common and one somewhat common microbe. And some of these might be familiar to you. They're on the left with that key with different colors. So Staphylococcus, people probably had staph infections, so very common microbe. Bacillus is common in the soil. It's very common everywhere too. And Fusarium and Aspergillus are both two very common fungi that we find basically everywhere. Dinococcus is not as common as those. It has been found in clean rooms, but it's a really incredible microbe that actually has shown the ability to repair its DNA when exposed to radiation. So they actually found film of Dinococcus growing on cameras that were inside 3 Mile Island because it was able to survive through that way. But what we did is we said, all right, well, for these five microbes, let's look at the experimental data where people tested what are the limits under which these microbes can survive exposure to ultraviolet light and to different temperatures. And then let's compare them to our UV environment. So how much ultraviolet light you get, which that little map on the top, those two little panels, they show the two big panels, sorry, they show how much light you get in different portions. So the brighter you are, the more ultraviolet light you get. The darker you are, the less ultraviolet light you get. Um, and then the bottom two big panels actually show you using this particular method, which is more of a rough estimate, what microbes might be able to survive in these different portions. And you might be like, hey, probably this is just a big splotch of pink. And you're right. It turns out that aspergillus, which is that's that same fungi that I showed you that was growing on the back of the wall and the ISS, it's really, really good at surviving ultraviolet light. And because the lunar South Pole never gets that hot, that's the main thing that ends up being the thing that kills a lot of these microbes. So Aspergillus, because it does so well, if it lands in a lot of these regions, it actually might be able to survive. Um, Now, these other colors, so for example, if you look at that orange with Fusarium, we have some zoomed in plots in the middle portion. So whenever there's a orange, that means not only can Fusarium survive, but so can Aspergillus. So basically, as you go up on these things, and you see one of those colors, if you see that color, all those other microbes can survive too. So the hardiest, so one, there are regions everywhere, all over the poles, based on this modeling, where these different microbes could survive. Some of these microbes are more hardy with respect to survival and may be able to survive in more of these kind of niches that exist in the lunar South Pole. So what we did is we tried to look at this and we said, that's great. That's this big global rough estimate for what might be able to survive. But how about the places we want to explore? So if you remember for Artemis 3, those yellow boxes that showed where we wanted to potentially land, and we wanted to potentially have a base camp, we said, let's do some really more sophisticated modeling. Let's basically break things down to smaller boulders and craters and try to figure out, if we start to do really complicated modeling, where can these things survive for at least one day? So you might be like, well, why do you care about only one day? So if you look at previous times that we explored the moon, once astronauts get to the moon, we don't just like get there and then sit around and watch TV. We're busy. So what they do is we say, all right, go out, do a traverse. So get outside, pick up rocks, run an experiment, put this thing on the surface, come back in, and then take a rest, and then go back out and do another one of those traverses. And the longest time between traverses has never been more than about 16 hours or something like that. I think it might be even shorter. So the interesting thing there is, let's say if I was an astronaut, I wanted to go out, and I know this place is basically like a giant freezer, which means it might have really interesting samples. If I wanted to pick up a sample, and I know Stacy, the other astronaut, just went out 12 hours ago, well, Stacy might have just left a microbe there, and if I scoop up that piece of soil and I bring it back, I now have to worry about, is that Stacy's microbe or is it something really interesting that I'd be very excited about? So that's one of the reasons why we care about this, but that's also why we cared about at least having one day as a survival limit. So if you go to less than one day, some of these things could probably survive as well. But here you can see in these three different regions that we highlighted that are all potential landing sites, every one of them has portions, and not just around the shadowed areas, but also in other areas where Aspergillus, that fungi, could potentially survive. If you zoom in, you can actually see other colors as well. So other microbes could also survive there. And these are just five that we picked. There's thousands upon thousands of other microbes that are potentially things that could be transferred because we see them all the time and because they might have properties for that for survival. And that's something our team is trying to look at in the future. And the reason why they can survive is because of this table on the bottom left. So this table kind of shows, the last column just shows which one of these have been found in those clean rooms. And it turns out every single one of them have been found in clean rooms. But the way it's really protectionism. It's really protection and repair mechanisms that enable these microbes to survive. So in some cases, it's having a big coat that protects itself. In other cases, it's having pigments that basically block out that UV light. Basically sunscreen for microbes. And in other cases, it's actually repair processes that repair things that might be damaged by things that are otherwise damaging it. So there's a lot of different ways in which these microbes are enable themselves to survive. So last thing I'll talk about on this slide as well is that the last set of kind of plots here on the right, they show not just one day. They show how many days does it take to potentially kill Aspergillus if it landed in a particular portion in this region. So instead of one day, we're now saying, all right, are there places where it survives for more than one day? And any place that you see that's bright yellow, it Aspergillus could survive, according to this model, for at least 7 days. And any place that's white never really receives direct sunlight, or so it doesn't receive direct ultraviolet light and it's super cold. And in those cases, you really need to start to think about, hey, how about reflected light and stuff like that? But the survival can be even longer in some of those cases. So some of the places we most want to explore, because they might have water, because they might have all these interesting things that tell us about the history of kind of the solar system and Earth, they also are the best places where microbes could survive. All right, so we've talked about how microbes can survive, why we're going to the moon. How do they get from inside your cabin or inside your spacesuit to the actual surface? So one is I kind of talked about the airlock. So if you have cabin air that is pumped into the airlock, and then your astronauts get in there, you close the door to the cabin, and then you slowly pump that air out. In the ISS, for example, a small portion of that air directly goes to the exterior. So all of a sudden, some of that air that contains those microbes, that contains all these other gooey organics, which are the food for the microbes, that's all going out to the surface. Then, of course, once you pumped it down, you can never really pump it down to a vacuum. So when you open that door, you get a little bit more that goes out there. And that doesn't count anything that might be clinging to your instruments or to your spacesuit. The other thing people don't realize is that when an astronaut's walking around, they are constantly having the air within their own spacesuit being filtered, because as they're moving around, they're generating heat, they're breathing out water vapor, they're breathing out CO2. And the spacesuits have been designed so that they basically scrub the air within the spacesuit and then kick that to the exterior as well. And certainly the new ones are planning to basically vent to the exterior as well. So there are plenty of ways where things that are inside your spacesuit or inside a cabin can get out to the lunar surface. So just kind of wrapping this up, and then hopefully there's some fun questions. So why is this important then? So hopefully I've convinced you that there's a pretty good chance we'll have hitchhikers that come with us, and that they'll probably get out to the surface. So one, there's both good things and bad things about this. And as a scientist, I want to explore the moon. It is an awesome place where we will learn a ton. It will be transformational in multiple ways for humanity. But this is something to consider in terms of how exactly we explore the moon. On one hand, though, just the fact that we will bring life, it will happen, and that some of it might survive is pretty amazing. It shows that resilience, that life can survive these crazy conditions. And hopefully you've seen some of this with respect to how life can survive in really extreme Earth environments. So now we're just kind of seeing this frontier expand where we really are getting confirmation that life is incredibly hardy, that it can survive in really amazing ways, and that life is incredibly diverse too, because that's what enables it to survive in some of these places. On the other side of on the other side, on the other side of the coin, it is thinking about, we want to make sure we're smartly exploring, or we're at least thinking about how we are transferring life. Like I gave in that Stacy the astronaut example, we do not want to go somewhere where we think there might be really interesting information in the ice or on the soil, and by mistake we leave a bunch of things on that particular sample that make it harder to understand what is going on. We also don't want to do that because for an area that might be especially fragile, we don't want to really change it in a way that might impact a number of things we might want to do in the future. So as a scientist, I think about, well, I don't want that life that, let's say I dropped on a rock and then Stacy comes back, picks up that rock and is like, give me my Nobel Prize. I just found life outside of Earth. And it turns out it was just a Prabal bug, a Prabal germ or something like that. So that's, I think from a scientist perspective, one of the things we'd want to guard against as well. The other thing is we also do want to think about if you have a sustainable presence, if you want to explore for a long period of time, what are the health and safety impacts if we are potentially transferring life, if we are leaving it there and it's now sitting there essentially in a freezer. It is important to remember, though. I really like the outdoors. I like hiking, and one of the principles people often think about is leave no trace. And I absolutely try to do that. But the reality is, you will always leave a trace. So when I go hiking, I might make sure I don't throw any garbage anywhere. I might make sure I try to put out a fire, make sure I do all these things that I'm supposed to be doing. But the reality is, when I walk through somewhere and I step on a twig and break it, and make a bunch of noise, that woodpecker that's sitting in the tree, I probably just woke it up from its sleep, from its normal cycle, and all of a sudden I've changed how it's going to interact with its environment. Or I might have stepped on, I don't know, some fox's favorite stick that it liked to play with or whatever else. Now, a lot of these are not ecologically important, but it just shows that regardless of uh, how well you try to stop affecting your local environment, you will inherently have some level at which you will leave a trace. Now, that does not mean that you should not try to smartly explore and think about how you are leaving those traces. And that's something we want to think about for the moon, not only because there's tons of really good science on the moon, but also when we want to explore an environment like Mars, where we really are excited about habitability, we want to make sure we're not messing up kind of the environment there and hurting our ability to conduct really groundbreaking science there. Last thing, last little bits I'll talk about. You kind of see this on Earth. So, for example, Death Valley, really extreme environments in a lot of ways. There's been evidence that microbes have survived tens of thousands of years in samples collected at Death Valley. So microbes can survive in really harsh conditions for really long periods of time. So things to remember as we bring these hitchhikers with us to the moon is that we will almost certainly impact our local environment. We will almost certainly transfer life, unintended life, to the lunar surface, and we will leave a trace. But understanding how we're doing that, tracking it over time, is really important. And experiments and research are really important in understanding this. So is understanding extreme portions on our Earth, because they give us a really good model for how we would explore places that are off Earth. And that's really all I have. But I'm happy to take any questions related to this topic and other ones related to exploration of the moon.